All right, this week we are looking at the lab on heredity and genetics. This is found on page 13 in your lab supplement. And if we look at our lab objectives here for the week, clicking on this link right here, you can see that there's a number of terms that we need to be familiar with. So lots of vocabulary in this week's lab. I recommend that you look at the back page of the lab for this week. I think it's on page 18. Yep. 17 and 18, there's a lot of terms here, and making sure that you understand the different terms would be great to make flashcards with, so you're really comfortable with all this different terminology related to inheritance. There's a lot of bolded terms throughout the reading as well that explain some of those terms in a little bit more detail. Highly recommend that you go through those. So we're going to look at the lab objectives today, and I prepared a PowerPoint going over some of the terminology. So this is located on Blackboard under Learning Materials. So let's get started. So objective number 1 on page 13 of your lab supplement talks about a chromosome set. This is an example of a karyotype. A karyotype is simply a display of all the chromosomes that we would find in the nucleus of our body, of all of our cells. And we have 46 total chromosomes, or 23 different pairs. So your first slide looks like this, and we can see we have a pair of chromosome 1, a pair of chromosome 2, and so on. Each of these came from one of our parents. So this one may have come from mom, and this one may have come from dad. They carry the same type of information, but maybe a little different form of that information. For example, if it was something like eye color, hair color, your dad may have dark hair, your mother may have lighter hair, so they both carry information about hair, but it might be a slightly different version of the color of that hair. And when we look at hair specifically, it's one of those traits that's coded for on multiple chromosomes and genes, so we'll talk more about that a little bit later. But just know that we have a pair of each of these chromosomes. One came from the mom from the egg, and from the dad in the sperm, and when they combined to form you, it was a zygote with one of each chromosome for a total of 46 chromosomes. So if we look at these chromosomes, we can see the chromosomes 1 through 22, even though they came from each parent, they look pretty similar in terms of length and banding patterns. These are the autosomes. We call them the body, or cells that are carried, that carry traits for most of the DNA in our bodies. So 1 through 22 we call autosomes. And if there's a gene on an autosome that is passed on that maybe carries a specific trait, we call that an autosomal linked trait. So 1 through 22 are the autosomes. They look like one another. The 23rd pair, in this case, is an X and a Y. These are called the sex chromosomes, and these chromosomes carry information about making someone male or female, so those body traits that determine male versus female. So the Y chromosome is a male chromosome, so the information that makes a male is carried on this very short Y chromosome. There's not a lot of other information on the Y chromosome other than the male traits. So the X chromosome, the other chromosome on the 23rd pair, is what makes a person a female, but it also has other traits on it we're going to determine and when we go through our Punnett square analysis. But if a person has a Y chromosome and an X chromosome, that makes that person a male because of those Y linked traits that make a male. If we look at the next example, we can see this individual does not have a Y chromosome. Instead, the 23rd pair is a pair of X chromosomes. So both of these are X chromosomes. One came from mom, one came from dad, and this person is a female. So when there's a pair of X chromosomes, that determines strictly female characteristics. But again, there's other traits on the X chromosome. You can see how large that chromosome is that are not related to sex, but they are sex-linked traits because they're on the X chromosome. So this is a chromosome set. We have a total of 46 chromosomes. 1 through 22 we call the autosomes, and the 23rd pair we call the sex chromosomes. So looking at this DNA, so chromosomes are just simply DNA. When a cell is in interphase and it's not actively dividing, the DNA is found in the nuclear membrane, as always, but it's in the form of chromatin. So chromatin is loosely bound DNA, kind of looks like spaghetti. But as that DNA coils up around these special proteins called histones, you don't need to know the name of that, but they tightly coil and form these structures called chromosomes. So here's a chromosome, same thing as chromatin, but it's just tightly coiled DNA. And when it's in the shape of an X like this, it means that this chromosome replicated, which means it made a copy of itself. So there's an exact copy on the other side here, and it's bound together at a central point called the centromere. So the centromere holds together these two copies of DNA, which are, one is a replicated version of the other. And we talked about DNA replication in our first lab, but this is the X version of a chromosome. But if we look at our chromosomes, typically they're not in that replicated form. We don't have an X version. We just have one single chromosome here and another chromosome here that's not replicated. But if it's in the X shape, it's still a chromosome, and each half is called a sister chromatid. So this is a sister chromatid, this is a sister chromatid. They're mirrors of one another, so they look like one another, carrying the same information, and they're bound at the centromere in the middle. So if we look at a pair of chromosomes, here's a set of chromosomes. Say the purple one came from mom, the green one came from dad. When it replicates, it makes a copy of itself. So this is a replicated chromosome, but we still see two chromosomes here. So there's two chromosomes here, except for these chromosomes are replicated and they have that X appearance to them. So whether a chromosome is in a single version or it's been replicated and has sister chromatids, there's some specific parts you need to know. The centromere is located somewhere along that chromosome, and it separates the chromosome into two different arms. The shorter arm we call the P arm, the longer arm we call the Q arm. So when you think of P as being short, think of P for petite. Someone who is petite is on the shorter side. So petite is the shorter arm of the chromosome, or the P arm. And this helps us identify different genes. So these banded portions on the chromosomes represent functional proteins. So it contains the genetic code, the specific sequence of nitrogen bases that codes for an amino acid, which will bond together. Different amino acids will bond together to form a protein. So these are called genes, and they code for functional proteins. So each of these banded structures here. So we use the P and Q arm to help us find specific genes. So the location of a gene on a chromosome we call a gene locus. So think of location and locus. So if I say a particular gene is on chromosome number 2 and it's on the Q arm, I know to look on this portion of that chromosome, on the Q arm, which is the longer arm of that chromosome. Telomeres are just the ends of chromosomes. That does not contain information for functional proteins, so we're not going to talk about the term telomere. But centromere, you need to know, is that little narrowing if we're looking at a single chromosome, or if we're looking at a chromosome that's been replicated, it's that point that attaches the two chromosomes together. And these two chromosomes, these two chromatids will separate during anaphase when the cell is dividing, during mitosis or meiosis. So we have a gene, which codes for a functional protein. We have a gene locus, which tells us the location of a gene on a chromosome. And then we have the P arm, which is the shorter arm, and the Q arm, which is the longer arm. So when these sister chromatids separate, they're no longer attached to one another. And when they're in a single version like this, like we see here, these we call daughter chromosomes. So looking back at our karyotype, each one of these is a daughter chromosome. So it's not connected to itself, to another copy of itself, as we see here and in this example. So these are sister chromatids, but once they separate in anaphase and we see them by themselves in the cell, it's still a chromosome, but we call it a daughter chromosome. So if we look at these chromosomes, so here's a pair of daughter chromosomes. They're not replicated. So let's say the blue one came from dad, this orangish pink one came from mom. On that chromosome, we said there are specific genes which carry information about a trait. but those traits can be slightly different. So different versions or forms of a gene we call alleles. So this allele is a dominant allele, which means we give it a capital letter, meaning that whatever this allele is dominant over this allele, which means it'll be more likely to be expressed in some form compared to the recessive allele, which we give a lowercase letter. So this individual, if we look at, remember we all have a pair of chromosomes, so let's say this is chromosome number 1 with different genes on it. The different forms of those genes might be different from mom and dad, but we're a combination of those two alleles. So the genotype, which means the combination of alleles that are inherited on chromosomes, there's a pair. So we have big P that came from this parent and big P that came from this parent. So we say that that individual is homozygous dominant for that allele because they have two capital letters. This pair of alleles, again, these are genes at a specific location, but the different forms of those genes are expressed as alleles. This person is little a, so we say they are homozygous recessive for this particular trait. So little a, the lower cases mean we're looking at a recessive allele. And this person has one of each. It has a capital and a small letter, so that has both a dominant and recessive allele for that trait. So we say they are heterozygous. Hetero means different. This is capital, this is lowercase, so that's heterozygous. So homozygous dominant is capital P, homozygous recessive is little a, and heterozygous is big B, little b. So again, those are dominant and recessive alleles. The dominant allele is always a capital letter. The recessive allele, we assign a lowercase letter. And genotype is the combination of alleles that we inherit from our parents. The only way to determine someone's genotype is to do genetic testing and look at the specific alleles that are inherited on their chromosomes. Phenotype, on the other hand, is the way someone looks based on the appearance, or based on the genetics that they inherit, on the alleles they inherit. So sometimes we can determine someone's genotype by looking at their phenotype, but that's not always the case, and we'll talk about that later when we look at our genetics problems. But in the case of hair color, eye color, skin color, there's multiple genes that combine together with different alleles from each parent that determine your individual hair color, eye color, or skin color. We call that polygenetic inheritance, when there's multiple genes across different chromosomes or the same chromosome that determine a specific trait. But regardless, the physical appearance of those traits we call phenotype. So genotype is the allele combinations that you inherit. So when we ask for genotype, in the genetics problems, we're going to be asking for the allele combination. So genotype is just described by these different letters: homozygous dominant, homozygous recessive, or heterozygous. But phenotype is the physical expression of those alleles. So there's different types of inheritance. When we take an individual that's homozygous dominant for a particular trait, like in this case with the pink flower, P stands for flower color, and the dominant color, which is represented by capital P, is pink. So little p is the recessive allele for flower color, and in this case we can see that the recessive allele is white. So if we have simple or complete dominance, it means that the big P is completely dominant over the little p. So any offspring that inherits these traits, in this case, if we combined a flower that was homozygous dominant for flower color being pink, and we cross that with a white flower that is homozygous recessive, making it white, we would have offspring that inherit one of each of these from the flowers, and they are all pink. But notice their genotype. So the phenotype, the physical trait we can see, is pink, but the genotype is big P, little p, making them heterozygous. But they're all pink because the capital P is completely dominant over the little p. So if the capital P is present in the genotype, it suppresses that recessive trait, the white flower color in this case. So the heterozygous individual carries the recessive allele, but it does not express that recessive allele in the phenotype. So we call, in complete dominance patterns of inheritance, we call a genetic carrier someone who carries the recessive allele but does not express that recessive allele in any shape or form. So here's an example of, let's say you have a particular disease that is carried. So these parents are each heterozygous. They carry one recessive allele and one dominant allele, and when they make sperm and egg, and that sperm and egg carries one or the other of these alleles in, because remember, sperm and egg are haploid, they only carry one of these chromosomes. So depending how those two combine, we might have a child that inherits the good allele from either parent when sperm and egg unite, and that child is unaffected. But if the children adopt or inherit one of those bad alleles, but the other good allele from the other parent, they're going to be unaffected, but they're going to be carriers again, which means they carry the recessive allele, but they have the good allele for their phenotype that is normal then. And then if a child would inherit both of these bad alleles from each parent, so the pink is considered the bad allele in this example, then you have a child that has inherited the two copies of the recessive allele, and then they have that particular trait or disease. So in complete dominance, again, a carrier has the dominant phenotype, but it is heterozygous, which means it's going to be one large letter, one little letter in terms of the alleles that were inherited. Codominance is when the heterozygous has both of the alleles, but both are expressed together. So in terms of blood type, this is an example of codominance. We have blood type allele A, blood type allele B, and if each is inherited by the offspring, the blood type of that parent is, or of that child, is blood type AB. So they inherit both alleles and they express both alleles, which is different than type A or type B or type O. Another example is incomplete dominance. Incomplete dominance is where the heterozygous individual does not look like the dominant individual. It's an intermediate form. So that's why we call it incomplete dominance, because the dominant allele is not completely dominant over the recessive allele. So here we see the dominant trait is curly hair, and the recessive trait with two little c's is straight hair. So if someone inherits one of each of those alleles from their parents, then they are heterozygous and they have an intermediate form of that hair, which is wavy hair. So that's an intermediate form. That's an example of incomplete dominance. And here's a cute little picture of a light bulb and an ice cream cone coming together to form that intermediate form. So Punnett square analysis is the rest of today's lab. You're going to look at the different videos going over the sample problems from the genetics worksheet. And basically we're taking an individual whose genotype is given in the problem. And remember, when they undergo meiosis, these alleles will separate, and the eggs, half the eggs will contain big A and the other half of the eggs will contain little a. And for a father that might be heterozygous, half of his sperm will be big A, half of his sperm will be little a. So in every example, you're going to be given information about the parents and what their genotypes might be. And you're just going to separate these alleles and put one allele possibility here, the other allele possibility here, and then you're going to cross those. So inside the Punnett square are the different zygotes that would result from the fertilization of this sperm with this egg, for example. When these two come together, we would get big A, big A. If this sperm would fertilize one of these eggs, we would have big A, little a. And if this sperm would fertilize this egg, we would get big A, little a. And if this sperm would fertilize this egg, we would get little a, little a. So we always put the capital letter first when we do Punnett square analysis. And this tells us what the probability is for these particular parents and the different egg and sperm combinations they're going to have and if they came together. So of the 4 here, we see that 25, or 1 out of 4, would be big A, big A. 2 out of 4, or 50%, would be big A, little a. And 1 out of 4, or 25%, would be little a, little a. So we could say that a couple has a 25% chance of passing along that recessive allele and maybe getting an affected individual if this is associated with some disease. So these are the zygotes formed by these different combinations of gametes that would be formed during meiosis. So this is homozygous dominant, these are heterozygous, and this is homozygous recessive. So you can spend the rest of the time then going over the genetics problems, and it's important that you understand the difference between phenotype and genotype and be able to make predictions given information about each of the parents.